Connaissance, recherche,
sciences,
et sciences mathématiques,
théorie,
physique,
philosophie,
conférence,
archéologie,
nature.
So my slides are in French, but I'll speak English because that's faster. You'll notice that it will be necessary. So I start at the beginning. In the beginning, there were the mists of time, and from these mists somehow came the frogs, salamanders, and caecilians. And there's no consensus on where to insert them into the generally accepted tree of living vertebrates, or living and fossil vertebrates. You are here, by the way. So the modern amphibians could be monophyletic and could be nested among the dysaurophoid temnospondyls, or could be the sister group to the lysaurophan lepospondylus. Alternatively, they could be polyphyletic, with the frogs and salamanders being dysaurophoid temnospondyls, and the caecilians being microsauran lepospondylus. As you can see here, the two monophyly hypotheses have different implications on the age of the divergence between the modern groups than the polyphyly hypothesis. I've made use of this fact to approach the question of the phylogenetic position of the modern amphibians indirectly by the implications of the divergent state on the phylogenetic relationships. I've got two papers out of this. We got. I've also approached the question directly by doing phylogenetic analysis on matrices that were already published and which we modified. We've also written a review paper which is now submitted, and I've started with my thesis co-supervisor Gerhard Steiner in Vienna, phylogenetic analysis of molecular data to supplement this. Basically, this is my thesis, my doctoral thesis. So, molecular attempts at molecular dating of the origin of this amphibia tend to give very old ages. For example, this is the age of this amphibia in the Devonian, according to this recent study. And this is the age of Ichthyostiga and, in general, the oldest limbed vertebrates. Similar things happen throughout this tree. For instance, the oldest crown group frogs, according to the study, have the age of the oldest known stem frogs, and indeed the oldest known amphibians. This does not fit the fossil record, but it happens to fit the polyphyly hypothesis, because according to the temnospondyl hypothesis, you would expect the origin of this amphibia somewhere here in the late Carboniferous, early Permian. Similarly, for the lepospondyle hypothesis, you'd expect late Carboniferous. But according to the polyphyly hypothesis, this down here is the divergence between caecilians on the one hand and frogs and salamanders on the other hand. And indeed, this date is within the confidence interval of this one, which is the origin of this amphibia, divergence between caecilians and frogs plus salamanders, according to the molecular dating study by Zhang and colleagues 2005. But such a molecular tree supporting the polyphyly hypothesis would contradict itself, because all recent phylogenetic analyses have found the lepospondylus to be more closely related to us, sorry, my nose is a bit closed, than to the temnospondyls, which would mean that if the modern amphibians are polyphyletic, they would have to be paraphyletic with respect to the amniotes. But almost all phylogenetic analyses, including all molecular ones, including the two I just mentioned, find this amphibia to be monophyletic with respect to amniota. So we have studies where the dates support the polyphyly hypothesis, while the topology of the same trees contradict it. So we used 3 methods to date the origin of this amphibia. The publication of 2 of them, paleontological dating, I'll explain what that is. Molecular dating came out in 2007 in Systematic Biology. It's a large paper of 11 online appendices, don't miss them. And the 3rd method calculates confidence interval on the first appearance of taxa in the fossil record. The paper on this came out in 2008 in Acta Paleontologica Palynica, which means it's free as in beer. In the first paper, we made a super tree and added to it all fossils of modern amphibians that we could, 223 species in all, this one for instance. And various tests show that all potential bias in this tree favors 2 old divergent states. So this is what a small part of the supertree looks like, with lots of fossil species in their phylogenetic and stratigraphic positions. And the dating works like this. For instance, the crown group of frogs here, its minimum age is given by the oldest member, simply. Discoglossus oxoniensis here. The maximum age is naturally less well constrained, but the fact that we have stem frogs in the lower Jurassic but no crown frogs suggests that there were no crown frogs, because there were, I would expect to find ecological equivalents if they were there. We tested the congruence of this tree with the stratigraphy, and we found that this congruence, which means the congruence between the order of cladogenesis and the order of appearance of clades as fossils, is much better than random. The probability that it is random is about 1 in 10,000, which means that the minimal divergent states in our supertree cannot be severely underestimated. Because the congruence is so good, we went on to suggest in the same paper new calibration points for molecular dating, some of them with maximum ages. And then we used those calibration points in our own molecular dating study, also in the same paper. We used the dataset of jungle colleagues, complete with mitochondrial genomes, the program rates, and we played around with lots of combinations of calibration points, internal and external to this amphibia, with and without maximal ages. And we get results like this, like these, which are a lot closer on average to what those in our supertree than those found by jungle colleagues. The use of maximum ages for calibration points is controversial in molecular dating. It's not done often, so an obvious question was, does it make any difference? Does it matter? Well, yes, if we don't use maximum ages on any calibration points, we get results that are very similar to those by jungle colleagues, while if we do, we get very close to what the fossils say. The second paper is based on this publication. This is a method that does not use phylogeny and answers the double question of how probable it is that the clade appeared how long before its first appearance in the fossil record. We fed into this method a list of 1,207 localities which have yielded less amphibian fossils and the stratigraphic positions of those localities. We scanned in the geological map of the world to get the relative surface of the sedimentary rocks of the relevant periods. We added 5 models of exponential growth, which differ in their assumptions on the vulnerability of less amphibians to mass extinction events. And these growth models require a starting date. For those, we took 2 from the 2 molecular papers which I just mentioned. So we get results like these. Number 6 over here is just to show that growth model is needed. So, number 5 assumes that less amphibians are immune to mass extinction events, and the other 4 differ in their assumptions on how susceptible they are to mass extinction events. And as you can see, they all stay within the Permian, and for the most part even the middle and late Permian. So we conclude from these two papers that there is indeed a monophyletic list amphibia which appeared in the Permian, so less than 300 million years ago. And reassuringly, the two methods that use the phylogeny agree with the method that does not use it. And the much-cited fundamental conflict between molecular and paleontological data on origination dates of taxa disappears if molecular dating is done with enough internal calibration points internal to the taxa in question and with enough maximum ages. The direct approach, all three groups of hypotheses have been supported by published phylogenetic analyses recently, and we looked at these three, the matrices of these three papers to find out why they give such different results. I must add that McGowan and Carroll found a very special version of the polyphily hypothesis, which differs a lot from the more classical one by Anderson and colleagues. Out of this we got two papers. The paper on McGowan's matrix came out in Contributions to Zoology 2 years ago, which means it's free. It has 51 pages, as you can see, because we documented all of our changes to the matrix in very fine print. Our work on the matrix by Anderson and colleagues came out last year in Evolutionary Biology, which means that, unfortunately, not even the supplementary information is free, even though that's where all the science is. The rest is just a 3-page reply to a review paper. So McGowan's paper was a description of these animals, which are known from the Middle Jurassic to the Pliocene. Amphibians are fairly modern aspect, but with a lot of special oddities. At the end of that paper, there's an analysis of their phylogenetic position based on a tiny matrix. And McGowan got these results. So the Sicilians, together with the Microsauran lepus bundles, form the sister group to the frogs, plus salamanders, plus albanopetrontids, and all of these together are nested within the Temnos bundles. This is unique, was before Carl's paper, and is due to the matrix, which contains, for instance, an all-zero ancestor, which was constructed, modeled after basal Temnos bundles like this one. So the outgroup of this analysis actually forms part of the in-group. There are things like a duplicated character in the matrix, plenty of contradictions of single cells to the descriptive literature, many decisions that were not explained, especially the limits between states of continuous characters. So an obvious question was, what happens if we fix this? Which means, which topology comes out if we use the same taxa and, as far as possible, the same characters, but fewer questionable codings of the matrix. So we replaced the all-zero ancestor by two real outgroups like this one, quantified state limits, ordered some characters, gave some a step matrix, and because the matrix is so tiny, we were able to check every single cell for accuracy. And we ended up changing 35% of them and got the Temnos bundle hypothesis. Very classical. List amphibians happily reunited with albanopetrontids inside the Temnos bundles, outside the lepus bundles. So there are Microsauran lepus bundles and Temnos bundles in this matrix, but no Lysarophin lepus bundles, even though Lysarophins and list amphibians had been found to be sister groups by a series of papers till 2002. So we added the only well-known Lysarophin, Brachytectus, to the matrix and got the lepus bundle hypothesis, like this. List amphibians and albanopetrontids next to Brachytectus, inside the lepus bundles, outside the Temnos bundles. Then this animal was published. It's a dystrophic Temnos bundle and was described as a stem batrachian, which means more closely related to related to the frogs and salamanders until the Sicilians. Here's the oldest Sicilian, oldest known. This means the Anderson colleagues who described this found the polyphyly hypothesis. Clearly, this animal is as relevant as Brachydactylus, so we added it to the matrix and still found the lepus bundle hypothesis. The entire rest of the tree, all of the tree stayed the same, basically. This is the paper that described Geobrachydactylus, and it contains a fairly large matrix, so we were not able to check every cell, but still we found lots of disagreements of single cells with the literature, applied the usual treatment, and got the lepus bundle hypothesis like this. So the closest thing to an amniote is here. The amphibians are over there in the corner, next to Brachydactylus, and kept inside the lepus bundles and outside the temnos bundles by comparatively high bootstrap and prima values. Geobrachydactylus is here, inside the temnos bundles. Currently, basically I just finished looking at the matrix by Wilton and Coates, 2007, which is the largest matrix on limbed vertebrate phylogeny published so far. It found the temnos bundle hypothesis. It also found lots of things like the paraphily of groups that had been thought to be well-established clades. How come? Because of the matrix, which contains many disagreements with the descriptive literature, many continuous characters with unclearly defined states. As usual, unfortunately, all characters were unordered. There are lots of correlated characters in the matrix. A very common case is that the presence or absence of each state of what should be a multi-state character was treated as a binary character in its own right. So, building on the work of Damien Germain, who I think is here in the room, or at least was recently, we made hundreds of changes to single cells in this matrix, ordered some characters, defined some state limits of continuous characters, and I have merged a lot of correlated characters, so the total number of parsimony informative characters has shrunk a lot. For separate analysis, I added Geobrachydactylus and a few other interesting taxa, and I got the lepus bundle hypothesis. So these are the original results by Wilton and Coates. List amphibians here among the temnos bundles, and to move them over into the lepus bundles requires 9 extra steps. I get this currently with less amphibians, at least close to Brachydactylus, inside the lepus bundles, and to move them over to the temnos bundles requires 10 extra steps. When I add Geobrachydactylus and others, the difference increases surprisingly. So on this part of my work, I can conclude that every cell in a matrix has an influence on the tree. Mistakes in the matrix lead to mistakes in the tree, mistakes of unpredictable kinds and magnitudes. Caution is advised in defining characters in order to avoid ending up with correlated ones. And typographic errors are inevitable, of course, in a matrix that contains hundreds or tens of thousands of cells. But typos are mistakes, and mistakes in the tree lead to mistakes in the mistakes in the matrix lead to mistakes in the tree. With our changes to published matrices, we see the lepus bundle hypothesis popping up everywhere. But of course, we don't expect this to have been the last word on the subject. For instance, it is very easy to identify fossils that need to be restudied, perhaps even redescribed. I hope to be able to do some of that work in my planned postdoc in Berlin. Together with Gerhard Steiner, I've started to work for molecular phylogenetic analysis of molecular data. I'm compiling sequences of from GenBank of recombination activating gene 1, which we have heard mentioned in a previous talk today. This is a nuclear gene because in the mitochondrial genomes that are usually used, there's some signs of saturation which would lead to long branch attraction, could change the results. This will distinguish between the two monophyly hypotheses and the polyphyly hypothesis. And as a side effect, it will tell us something about the phylogenetic positions of the turtles, which is the other great controversy in the phylogeny of the limbed vertebrates. And a surprise on this topic just came out a few days ago. And that's it. Thank you.